Hello everyone, it's good to be back again to share some things about biology with you all. Today we'll do revision of Form 4 Biology, Chapter 3, the movement of substances across the plasma membrane. We will try some common examination questions as this is the best way to get prepared to ace our examinations. Before we begin, let us revise the osmosis rule again. Water always diffuses or moves by osmosis from a hypotonic or dilute solution to a hypertonic or more concentrated solution. Okay, so you have to memorize this rule: hypotonic to hypertonic or dilute to concentrated. So you must know what's the meaning of the word hypotonic. A hypotonic solution is a solution that is more dilute compared to another solution. And more dilute means it has a higher concentration of water molecules or higher water potential than another solution, or it has lower solute concentration compared to another solution. Okay, so the concentration of water, if it's higher, that means the solute concentration of the same solution will be lower. It's like it's inversely proportional. Anyway, just remember this rule. From hypotonic solution, the water will diffuse by osmosis to hypertonic solution, or from dilute to a more concentrated solution. Okay, so if you have not written this rule down, you can write it down somewhere in your notebook. Let's look at question 1 now. The diagram shows an experiment which was carried out to study the movement of substances across selectively permeable membranes. What is the outcome after 30 minutes? Okay, so here is a potato that's peeled, and then a part of it was cut out, and to produce a cavity here, distilled water was filled into here, and the whole potato was immersed in 30% sucrose solution in a petri dish. So you can pause the video and think about this question and try to answer the question for a while. And we'll come back to this in a short while. Try and get the answer by using the principles of osmosis. Okay? Right, did you get the answer or an answer? Okay, let's see how we can solve this question, this problem here. Now, first of all, in osmosis, there will be two solutions, like distilled water and 30% sucrose solution. One will be the hypotonic solution compared to the other, and the other will be the hypertonic solution compared to the first one. So between distilled water and sucrose solution, 30% sucrose solution, which one is the hypotonic solution? Yes, it's distilled water. Hypotonic solution or dilute solution, and it has a higher concentration of water molecules. In fact, the concentration of water molecules here for distilled water is 100%. It's 100% water molecules. Okay, and the sucrose solution, it is 30% sucrose solution, meaning 30 grams of sucrose dissolved in 100 ml of water. So it's very concentrated. So this is the hypertonic solution because it has a higher concentration of solutes. Okay, right, the concentrated solution. Then the potato has another part that's involved in osmosis, and that's the semi-permeable membranes. So the potato contains a lot of cells, potato cells, and each one has the plasma membrane, which is the semi-permeable membrane. Okay, now you're ready to answer the question. According to the osmosis rule, water always diffuses by osmosis from the hypotonic solution to the hypertonic solution, from the more dilute solution to the more concentrated solution. Okay, so we can use an arrow, we can draw it in the diagram to show the direction of diffusion of water by osmosis. So you will have to draw your arrow going out from the potato into the solution. Now once you draw this arrow, it's clearer to see the result, for you to see the result. Okay, then you can answer the question. Now, since water diffuses out from the distilled water into the sucrose, 30% sucrose solution, that means the distilled water is losing the water molecules, so the level of distilled water will go down. But the level of sucrose solution will come up because it's receiving the water molecules. Okay, so let's look at the answers and see which one is the correct one. So this is the original question. So which one is the answer? The level of sucrose solution in the Petri dish decreases. Is wrong. Okay, it should be increased. Level of water in the cavity decreases. Yes, this is the correct answer. So B is the answer. Okay? And C and D, if you read through, they are not the answers. B is the answer. Therefore, when answering questions on osmosis, always try to determine the hypo or hypertonic solutions first, because there will always be two solutions involved, and you may have to find out where is the semi-permeable membrane. Secondly, the direction of movement of water, where does it go? And we must follow the osmosis rule, that is, water always diffuses from the hypotonic or dilute solution to the hypertonic or more concentrated solution. Lastly, you may be asked about the effect of the flow of water or diffusion of water on the cells or tissues or whisking tubing. Let us discuss question 2 now. The diagram shows a cell which has been immersed in a sucrose solution X for 20 minutes. Which of the following could be the concentration of solution X, and what is the condition of the cell? So you have to refer to this diagram here, analyze it, and then try to find the answer here. So you can pause the video for a while and then try to find the answer. Okay, now let's look at the cell. It shows the cell, a cell that has undergone a process called plasmolysis, right? Where it has been placed in a hypertonic solution, which is more concentrated than the cell sap of the plant. So water diffuses out by osmosis, out of the cell, cytoplasm shrinks, vacuole also shrinks. This causes the plasma membrane to pull away from the cell wall. Okay, so this curved line here, all these curved lines are the plasma membrane, which is no longer together with the cell wall here. As the cytoplasm shrinks, it has pulled away from the cell wall. But the cell wall is rigid, so the shape is maintained here. Okay. So this cell has undergone plasmolysis. As we can see here, which one has undergone plasmolysis? So it's either A or C answer. And we must find a hypertonic solution to the cell sap to represent the concentration of X. Okay. So which one is an isotonic solution? First of all, find out the isotonic solution first. So you must know the isotonic solution by heart, and it is 5% sucrose solution for plant cells. This is the isotonic solution for plant cells. 5% sucrose solution means dissolving 5 grams of sucrose in 100 ml of water. Okay. The textbook gives the value in mole or molar. Molar means moles per 1000 ml, okay, of distilled water. So 0.5 m. So these two values must be memorized because it is the isotonic solution for the cell sap that when compared to the cell sap of plant cells. Now, if this is an isotonic solution, that means 10% and 15% are the hypertonic solutions. 10% means 10 grams of sucrose dissolved in 100 ml of water. Okay, so out of these two answers, for 15%, third digit is wrong. It's out, so it is C. The answer is C. Okay, hypertonic solution causes plasmolysis of the cell. Now, number 3, identify structure F and the substance in G. As we said, F is the plasma membrane, right? So it must be D. But what about G? So when we immerse the plant cell in a sucrose solution, the sucrose solution can move through the cell wall here, because the cell wall is not like the plasma membrane. Plasma membrane is selectively permeable or semi-permeable, but cell wall is fully permeable. So the sucrose solution can pass through the large pores in the cell wall, and it will fill up the space here, all the white spaces here, right? It's filled up with the sucrose solution from outside. So the answer is D. Let us discuss question 4. Molecule R passes through the plasma membrane by using the transport protein shown. What are the characteristics of R? Now take a moment and pause the video and then try to find the answer yourself, and also go through number 5 and try to answer the question. So pause the video even right now if you want to, and try to get the answer yourself. Right, so coming back to the question number 4, let's look at this picture. It shows a transport protein transporting molecule R through it. So there are only two types of transport proteins: channel protein and carrier protein. Is this a channel protein? No, because there's no straight tunnel here. So it's most probably a carrier protein. Now, is it the carrier protein for active transport or passive transport? It's for passive transport, because you don't see an ATP molecule here, okay, breaking down. And also the substances, that means R, is transported from region of higher concentration, there are 3 molecules here, to region of lower concentration, only 1 molecule here. So it's passive transport using carrier protein. And the type of passive transport must be facilitated diffusion, okay, using carrier protein. So R is either glucose or amino acids or some other large polar molecules, okay, that cannot pass through the hydrophobic tails here, because hydrophobic tails are nonpolar. So they use the glucose molecules and amino acid molecules will use the carrier proteins, which can change shape to transport them across the plasma membrane. So answer is A, large polar molecules. Number 5, which of the following processes do not require energy? So these processes that do not require energy are the passive transport processes, involve passive transport. Okay, so for passive transport, there are 3 types. Think of the word SOFA, SOFA, our acronym. The 3 types of passive transport or movement of substances across plasma membrane are S, O, and F, which is simple diffusion, O for osmosis, and F for facilitated diffusion. Then the last one, A, is active transport. So let's find out which one is a form of passive transport. Okay, absorption of glucose in the villus. So I like to clarify here, because there are many questions from students about this. Is this by active transport or facilitated diffusion? Now, especially for absorption of glucose in the villus, we consider this as taking place by active transport. Okay, although there are examples of facilitated diffusion using where the glucose molecule is transported by facilitated diffusion using carrier protein, okay, into the cell, to be transported into the cell. So it can be transported by facilitated diffusion also of glucose, which is a form of passive transport. But when you see the absorption of glucose in the villus, then it's active transport. Special case, okay? Right. Reabsorption of glucose in the kidney, active transport. Transport of sucrose from leaf to phylum tissue, active transport. Absorption of fructose molecule in villus, passive transport. This is by facilitated diffusion, which is a form of passive transport. Fructose is not the same as glucose. It's a different molecule, although both are sugar molecules. Okay? So D is your answer. By facilitated diffusion. So it does not require any energy to transport the fructose molecule across the plasma membrane. Let us go on to question number 6. A student used a microscope to observe some red blood cells that had been immersed in a saline solution R for 10 minutes. He drew the cells as seen in the diagram. Which of the following inferences can be made from his observations? The cells have lysed or burst. B. 1m saline solution will produce the same result as R. C. The cells are plasmolysed and flaccid. D. 0.1 m saline solution has the same effect as R. Okay? Think a while, a moment to try to get the answer yourself. Okay, now looking at the cells here, the red blood cells, we can see that they form the star-shaped structure like this, and this shows that they have undergone crenation. Okay, so crenation happens when the red blood cell is immersed in a hypertonic solution, causing water to diffuse out by osmosis, and when the red blood cell loses water, it will shrink or shrivel, right? So the solution outside must be hypertonic solution to the red blood cell. So how do we find out the type of solution used? For example, here they say 1m saline solution. Okay, so you must know the isotonic solution first, the one that is isotonic to the red blood cell. What is the concentration of the saline solution that is isotonic to red blood cell? Okay, right, we start with the first choice for answer. The cells have lysed or undergo hemolysis, or they have burst. Not true. Okay, hemolysis or lysis only happens in hypotonic solution, when the solution is more dilute than the cytoplasmic fluid of the red blood cell. But in this case, we want to look for a more concentrated solution. Okay, so this is definitely wrong. Now, B, 1m saline solution will produce the same result as R. So is 1m saline solution a hypertonic solution? All right, so referring back to our isotonic solution for red blood cell is 0.15 molar molar saline solution or salt solution or sodium chloride solution. So compare 0.15 m with 1m. Definitely 1m is higher in concentration than 0.15 m. Okay, so it's a hypertonic solution, and it will give this result that we see here. So B is the answer. Now let's look at C and D. Cells are plasmolysed and flaccid. We cannot use the term plasmolysed or flaccid for animal cells or human cells. This is for plant cell. Okay. D, 0.1 m saline solution has the same effect as R. 0.1 m saline solution compared with 0.15 m saline. This solution, 0.1 m, is around this value. Alright, so it's either slightly hypotonic or isotonic, like this solution, this 0.15 m solution. Okay, so it isn't hypertonic. So D is not the answer. Okay, now lastly, you have to study this table and make sure you know all the values here. For red blood cell, the isotonic solution is 0.15 m saline solution or sodium chloride solution or salt solution. Okay, molar, this one is the molar, means 0.15 moles of sodium chloride dissolved in 1000 ml of water. Okay, 0.9% means 0.9 grams of sodium chloride salt dissolved in 100 ml of water. For plant cell, the isotonic solution is the sucrose solution, 0.5 m sucrose solution, and in terms of percentage is 5% sucrose solution. Okay, so memorize that. Now let's look at a HOTS question, which is a subjective question, either an essay or structured question. Okay, to test your thinking skills. Question 1. The diagram below shows two processes, X and Y, which are carried out by root hair cells to help them obtain their requirements. A. Based on the diagram below, compare and contrast the two processes, X and Y. Okay. And secondly, what happens if a respiratory poison is added to the root hair cells? Okay, so here we see the root hair cell, what of it? And we see two arrows showing that water is going into the cell and also nitrate ions. And water goes in by process X. So what is process X? Yes, it must be diffusion, because water is taken in by the plant by the process of diffusion of water is called osmosis, right? So X is osmosis. And for Y, nitrate ions are taken in by what process? Nitrate ions are a type of mineral ions, so it's taken in by active transport. All right, these two are different. These two types of transport are different. For water, it's by osmosis, and for nitrate ions, it's by active transport. Thus, we must write down that X is osmosis, write it down first, and Y is active transport, so that we do not get confused later on. Okay? Right. So coming to the compare and contrast command word, we have to give some similarities and some differences. It cannot be totally all differences or all similarities. You will not be able to get the full marks, because marks are given for similarities and for differences. Right, so we're going to compare osmosis and active transport. Basically, osmosis is passive transport, okay, and Y is active transport. So think about the differences between passive transport and active transport. And then of course also the similarities. Okay, we start with the similarities. So both processes involve the movement of substances from one area to the other. For example, water is moved into the cell, and nitrate ions are moved into the cell. And then both processes here involve a selectively permeable membrane, and that is the plasma membrane. So the substances will move in through the plasma membrane, which is a selectively permeable membrane. Thirdly, both processes are needed for the growth of the plant. Now, coming to the differences, you can pause the video for a while and just take out a piece of paper and try to think of the differences between osmosis and active transport. So pause the video for a while and try to think what are the differences first. Then we'll discuss it here. Okay, now in the last few videos, we have discussed the characteristics of osmosis and active transport. And we know that osmosis is a passive process, okay? Whereas active transport involves the use of energy, which osmosis does not, as a passive process. So the first difference is that osmosis is the movement of water molecules, the net movement of water molecules, from an area of high concentration to an area of low concentration. But this active transport is different. It's the movement of the nitrate ions from a region of low concentration to that of high concentration. Okay? And so we can say that for osmosis, water molecules move down the concentration gradient, whereas in process Y, the nitrate ions are taken into the root hair cells, but they move against the concentration gradient. That means inside the cell there's already a lot of nitrate ions, yet they want to move it into the cell by active transport. Okay, the third point is that for osmosis, no energy is needed because it's passive transport. But for active transport, energy is needed. And what does it result in? For osmosis, it results in the dynamic equilibrium where molecules of water are evenly dispersed in the medium. For process Y, it results in accumulation of the nitrate ions in the cell because the nitrate ions are taken in, and even though the cell has a lot of the ions already. Okay, so there's an accumulation of the nitrate ions and there's no dynamic equilibrium. And lastly, it does not involve a carrier protein. This is for osmosis because if it occurs in the cell, the water molecules will not pass through any carrier protein because it's very tiny. You can just squeeze into the pores of the phospholipid bilayer and move through it. Okay, but for Y, it must involve a carrier protein that can change shape. Okay, so in this case of the nitrate ion, since it's transported into the cell by active transport, it must involve a carrier protein. The last question is, what would happen if a respiratory poison was added to the cells? So if a respiratory poison was added to the cells, process Y would stop. Active transport would stop. Because the respiratory poison is actually a poison that will stop cellular respiration in the cell. Okay, so the root hair cells actually require energy from ATP, from the breakdown of ATP, to carry out active transport, which is process Y. And to produce this ATP, the cell has to carry out cellular respiration, and then that will provide the energy which is used to synthesize the ATP. So now the respiratory poison has stopped cellular respiration. So there's no cellular respiration, that means there's no energy produced to synthesize the ATP. And once that happens, there's no more ATP in the cell, and it cannot provide energy to the root hair cells for active transport. Okay, so no energy is produced to synthesize ATP for active transport. Okay, no energy from respiration is available to synthesize ATP from active transport. The cells will not have any energy to carry out active transport. Okay,